This is Dr. Morgan. I'm going to be recording some lectures. This first one is basically chapter 19, blood or hematology. This first exam is the overview of blood. Remember, blood is a fluid connective tissue. It represents about 8% of your total body weight. Circulates throughout the blood at all times. And when you take your blood from, let's say, from the body, you'll get what we call a peripheral blood sample, and it can be spun down. And once it's spun down or centrifuged, the blood will separate into its individual components. One of these components is called the plasma. It's basically what we refer to as the liquid extracellular matrix of the blood. This area right here that you see. But as you can see in the inset, the plasma represents water, plasma proteins, and a few solutes like glucose, electrolytes, and minerals. Okay. Now, 1% of your peripheral blood will consist of this area here that we call the buffy coat. Buffy coat is basically cells, basically white blood cells and platelets. The other name for white blood cells are leukocytes. It's the leukocytes that are our body's cells that basically are defensive mechanisms for the human body. In other words, when we have an infection, it's the leukocytes that basically put up the defense for us. And we will look at the leukocytes and distinguish the different types of leukocytes later when we get to the discussion on leukocytes. And then the third portion that you see on your blood, your centrifuged blood, are what we call the red blood cells, which are also called erythrocytes. It represents about 44% on average of your total blood volume, and that's what we refer to as the hematocrit. Hematocrit, by definition, is the percent red blood cells per unit volume. It averages about 44%, but this value will actually vary between the genders. Males, because of their increased body mass, will have a little bit higher hematocrit than what you would see in females. Let's go to the next slide. So let's look at the three types of formed elements that are formed primarily in your bone marrow. Okay, your most common of your blood cells are referred to as the erythrocytes, also referred to as red blood cells, primarily because of their noted color. They stain a bright red primarily because of the protein hemoglobin, which carries on a red color when hemoglobin binds with oxygen. The second formed element, which is also made in the bone marrow, are the leukocytes. Leukocytes are also called white blood cells. Like I said before, white blood cells are part of our immunodefensive mechanisms of the body. So they're very important in keeping us disease-free. Platelets, now the platelets, the third type of formed element, are actually fragments of a particular type of leukocyte called a megakaryocyte or macrophage. But remember, platelets are very important for blood clotting. And without platelets, of course, we would have extended bleeding times, and some people would bleed to death if the platelet count was extremely low. So the three distinct layers that are formed in your blood sample once it's centrifuged is basically the plasma, which is about 55% of the total volume. It's going to separate as the top layer because since it's mostly water and proteins and small solutes, it'll separate out on top. But it represents the largest percent of your blood sample. The middle layer, which is basically sometimes referred to as the buffy coat, is basically going to be composed of leukocytes and platelets. And remember, it only represents 1% of your total blood volume. So it'll separate out in the middle because its density is much less than the erythrocytes, but a little bit more than the plasma. And then the bottom layer, which is going to be representing 44% of your total volume, is basically your erythrocytes or red blood cells. And basically the percentage of red blood cells by volume is what we refer to as the hematocrit. So basically, this is a non-centrifuge blood sample on the left here. So it's got this uniform suspension, which basically is going to be red in color, primarily because it's a heterogeneous mixture, but it looks red because of the predominance of the red blood cells. But after you centrifuge the blood sample with high-speed centrifuges, the blood will separate out into its three specific main components, which are your plasma, buffy coat, and then finally the erythrocytes. So let's look at some of the functions of blood. Of course, blood is primarily involved in gas exchange, exchanging both oxygen, carbon dioxide, and then transporting that into the blood. It basically transports many of your ions, it distributes solutes like ions and nutrients and hormones, waste products, and also plays a big role in regulating your electrolytes in your tissues, your ion concentrations. And then the third function, which is attributed to mainly the leukocytes and proteins, are your immune functions. Okay, and these are basically referred to as the leukocytes and the immunoglobulins or antibodies, also called the immune system proteins, which are transported throughout the body in the blood. Remember, these are going to play a very big role in fighting infection. And since I'm doing this lecture primarily because of the COVID-19 health scare, health crisis scare that we've had recently, this is going to be very important to understand, especially in light of the activities of today. So let's look at some of the other functions of your blood. One, it maintains your body temperature. Remember, blood will carry away heat, which is generated as a byproduct of many of the chemical reactions in the human body, and it functions in coagulation, blood clotting. It helps you seal damaged blood vessels, prevent any kind of blood loss. Third function is basically it preserves homeostasis by keeping your acid-base homeostasis in check, in balance. Your pH of your blood is maintained at all times between 7.35 and 7.45. That's only 0.1 pH unit difference. Okay. And that's relatively constant, and that's because you have several buffering systems that are at work in your blood. And the last function would be stabilize your blood pressure, mainly through adjusting the blood volume. Remember, your blood volume is a major determinant in your blood pressure. Let's look at the plasma and more specifically. Remember, your plasma is about 90% water, and it's the amount of water that determines how thick or how viscous the blood is. So that's what we refer to as the viscosity. So if you have more water, that means it's less viscous, and if you have less water, it has more viscosity or thickness. People that have let's say increased red blood cell production, there's a condition called polycythemia, where the blood cell production is in excess of what it should be, in excess of that, that 44%, and that can cause havoc with your blood pressure because of the increased viscosity. Remember, less water leads to greater viscosity and more sluggish blood flow. So it influences circulation a great deal when the viscosity is not constant. Also present in the plasma are your plasma proteins, which form what we call a colloid. Remember, colloids are aggregate proteins that are important in forming certain solutions. They make up about 9% of your total plasma volume. Albumin is your main colloid. It's a large protein that's only synthesized in the liver, and it's the main protein that's responsible for your colloid osmotic pressure, which always draws water into the blood by osmosis. Remember, with albumin, basically your colloid osmotic pressure is fairly constant. And then also we also have immunoglobulins, or immune proteins, also called antibodies, which are made by many of your leukocytes, which are a very important part of your immune system. These are also present in the plasma. Of course, they're transported to your tissues, and of course they're going to be important in fighting infection. Another type of plasma protein that's, of course, most of these proteins are made in the liver. Another transport protein are your lipid-based molecules, like your lipoproteins that are important in carrying cholesterol. So they're usually going to bind to lipid-based molecules, some of these transport proteins. Otherwise, they would be incompatible with your water-based plasma. And it allows transport of many of these lipid-based transport proteins in your blood. Very low density lipoproteins and low density lipoproteins and high density lipoproteins. These are a few examples of lipid-based transport proteins that carry cholesterol in your body. Clotting proteins. Clotting proteins, which form part of our coagulation cascade, these are very important for controlling bleeding, stopping a person from bleeding when an injured blood vessel forms a blood clot, with assistance also from the platelets. So the combination of platelets and coagulation proteins, or clotting proteins, the blood will basically coagulate on the average of about 1 to 3 minutes. And then the last 1% of your plasma volume are primarily your small molecules that are dissolved in solution, which form most of your electrolytes, like your sodium and potassium and calcium and so forth. Those molecules are generally readily exchanged between the blood and the interstitial fluid in your capillary beds. So this table, 19.1, basically gives you kind of a list of all of these components of your plasma, their approximate percentage, and then their functions within the plasma. Liver cirrhosis, or liver disease. You have many causes of liver disease. By far the most common cause is alcoholic liver disease. Other causes include cancer, which many are associated by genetic predisposition. Some are associated with hepatitis C viruses, alcoholism, like we mentioned, and viral hepatitis. These all can cause liver disease or cirrhosis. Once liver disease or cirrhosis starts occurring, it usually is a non-reversible event. In other words, it's going to keep on going. It basically can't regenerate after you reach the liver cirrhosis point. It's very common in the United States today. It's the 10th leading cause of death for men and 12th leading cause of death for women. And one of the principal problems that occurs in the human body is a decrease in plasma proteins, in the production of plasma proteins. So you'll get a blood smear and a complete blood count, you'll get a metabolic panel, and you'll find out that the colloid osmotic pressure is reduced because the albumin level is usually reduced. And that results in a lot of fluid loss to the extracellular space, and that can produce severe edema, sometimes in the abdomen, and that term for edema in the abdominal area in the peritoneal cavity is called ascites. I've seen cases where the fluid in the peritoneal cavity has to be drained just to assist them in breathing. And also you have clotting factor declines, and these also cause easy bruising and delays in clotting. This, of course, can be fatal if it's not corrected. Okay, the last function, erythrocyte and oxygen transport. Of course, your red blood cells are basically biconcave dish-shaped, and that form is very important because it helps the red blood cell transport, over 1 million hemoglobin molecules that it carries to the different tissues for oxygen exchange with the tissues. It's a biconcave disc which resembles a flat donut-shaped cell because it lacks a nucleus. And that donut shape also assists in the transport of hemoglobin because it leaves more room, since it doesn't have a nucleus, for the transport of hemoglobin molecules. So this is an image, electron micrograph scan, elevated, magnified about 4200x. And this is basically that biconcave disc shape of a red blood cell. So that surface area for the red blood cell shape is very important for its chief function of your blood, which of course, like I mentioned, is gas exchange. Mature red blood cells are anucleate, which means they lack their nucleus, and they also lack most of their cellular organelles. So once the red blood cells have produced all the hemoglobin that it's going to produce, which is is basically only going to occur in the bone marrow, then it's really not necessary to have another nucleus or a nucleus that persists for the lifetime of the cell. And it also lacks most of the other cellular organelles like the mitochondria and the Golgi apparatus and many of the ribosomes. Okay, so it doesn't have any means of repair. Okay, so red blood cells, since they are anucleate, they're not going to live for but about 4 months. So they have to be completely degraded mainly by the liver and the spleen, and then more red blood cells made every 3 to 4 months. And that anucleate form also helps the red blood cell create room in the cytosol for many of the enzymes and the nearly 1 billion hemoglobin proteins that carry the oxygen to the tissues. So here's your red blood cell shape, your erythrocyte structure, that biconcave disc shape, and these are the hemoglobin molecules within the red blood cell. With various enzymes also scattered in the erythrocyte. The oxygen molecules are the light orange circles that you see here. These are the light orange discs. And then dark orange is the hemoglobin that doesn't have a iron, or the iron doesn't have an oxygen attached to it. So this would have 3 oxygen molecules, so that means this hemoglobin molecule is 75% saturated. A maximum loading up of oxygen would be 4 oxygen molecules, which that means that hemoglobin molecule would be 100% saturated with oxygen. So hemoglobin, let's talk a little bit about the hemoglobin molecule. It basically has 4 polypeptide subunits, 2 alpha chains and 2 beta chains. And each of these polypeptide chains will also have an iron-containing compound called a heme group. It's a heme group that's instrumental in carrying the oxygen. When that iron ion in each heme group is oxidized, it binds to the oxygen, especially in regions of high oxygen concentration like the lungs, and then it forms a red molecule referred to as oxyhemoglobin. And this is, of course, just what's carried eventually to the tissues. You can buy at Walgreens or CVC, these pulse oximeters, and kind of get an estimate of your percent saturation. And that basically is reflecting the amount of oxyhemoglobin present in your body and the saturation. So anyway, these are your 4 polypeptide chains, each with a heme group attached in the middle of each polypeptide chain. 2 alpha chains and then 2 beta chains. The dark orange and the light beige color is basically the 2 alpha chains. And the hemoglobin, which carries only the oxygen molecules via the iron molecules, is basically right in the middle of each of these polypeptide chains. Now, hemoglobin then will release the oxygen into regions where the oxygen concentration is low, like in your body tissues. And also it can bind, hemoglobin can also combine to carbon dioxide, in which it forms carbaminohemoglobin. It usually does that where the oxygen levels are low. About 23% of your carbon dioxide is actually transported in the blood as carbaminohemoglobin. And we'll talk about the rest of the carbon dioxide transport later when we look at the bicarbonate levels and the dissolved CO2 levels in the solute. But 23% is actually carried as carbaminohemoglobin. Unfortunately, hemoglobin can also bind to carbon monoxide, and when it does that, it forms carboxyhemoglobin. Carbon monoxide has a very strong affinity for the hemoglobin molecule to the iron atom. It's almost 200 times more stronger affinity than what oxygen has toward the iron atom in the hemoglobin. And what that does is when that carbon monoxide binds to this hemoglobin, it changes the shape of the hemoglobin. And that then makes it unable to unload the oxygen into the oxygen-deprived tissues. And that's what leads to death, the asphyxia. So this is the oxygen molecule that's bound to the heme group, which specifically is the iron atom. And that's what we term oxyhemoglobin. This is a heme group in deoxyhemoglobin. As you can see, there's no oxygen molecule bound to the iron atom here. So this is what you see mainly in a high concentration in your venous blood. And this is what you would see in high concentration in your arterial blood. Your red blood cells don't live a tremendous amount of time. They live for about 100 to 120 days, about 3 to 4 months. And that's primarily because of the damage that they incur as they go through the different tissues and supplying the tissues with oxygen. And also remember they lack any means for self-repair. Remember they've already lost the majority of their organelles by replacing that cytosol with hemoglobin during their maturation process. So your body has to continuously make new red blood cells. So the process of making new red blood cells is what we refer to as hematopoiesis. Of course, it always occurs in the red bone marrow, and that's where your formed elements in your blood are produced by your hematopoietic stem cells. Erythropoiesis is a specific process of hematopoiesis that produces only red blood cells or erythrocytes from these hematopoietic stem cells. It takes about 1 week, 5 to 7 days for erythropoiesis to occur. It begins initially when the stem cell differentiates into a progenitor cell, and these are referred to as erythrocyte colony forming units. And at that point, they're committed to forming only one cell type or one colony forming unit. The erythrocyte CFUs then differentiate into what we call proerythroblasts only when the hormone called erythropoietin, which we just generally abbreviate EPO, when it's secreted by the kidneys is present. So your kidneys, as we'll see later, are the organ that regulates your red blood cell production whenever they experience any kind of tissue hypoxia or lack of oxygen. So here's your red bone marrow where your erythropoiesis is occurring. And here's your stem cell, hematopoietic stem cell. It then becomes any type of formed element. In this case, it becomes erythrocyte, colony forming unit. It's now committed to becoming this red blood cell. Then if the erythropoietin is present, then it becomes a proerythroblast. Proerythroblasts then further develop into erythroblasts. Blast, remember, means to build or to grow and develop. And these are the erythroblasts that rapidly synthesize hemoglobin, other proteins for carrying oxygen. At the same time, the nucleus in the erythroblast will shrink as it matures. Eventually, it's ejected from the bone marrow, resulting in a reticulocyte, which are what we refer to as the hollow donuts that we saw in the peripheral blood smear a while ago. Reticulocytes then enter the bloodstream after ejecting the remaining organelles in the cytosol. They do that by exiting through the pores in the sinusoidal capillaries of the bone marrow in the medullary cavity. So your early erythroblast is where the hemoglobin is rapidly synthesized, and then they become proerythroblasts, which then eventually become erythroblasts and then erythrocytes. So let's look at more detail at how erythropoiesis is regulated. Remember, erythropoietin is the hormone produced by the kidneys that triggers a negative feedback loop. So let's go through the feedback loops. Remember, a stimulus for producing hematopoietin is basically when the blood levels of oxygen fall below their normal levels. That's called tissue hypoxia. The kidney cells are the receptors that detect this falling oxygen levels, which is basically called tissue hypoxia, and then that incites the kidneys to produce more erythropoietin, and that hormone is introduced to the bloodstream. So you're going to produce more red blood cells. That your red cell or erythrocyte will increase. That becomes your response, and your blood levels, because your red cells carry more hemoglobin and more oxygen, the blood levels of oxygen will rise to normal. Oxygen levels go back to normal, and that inhibits any more production of erythropoietin, and that will basically close the feedback loop. So that's basically how erythropoiesis occurs, step by step. First, your kidneys experience tissue hypoxia. PO2 levels drop below the normal range. Kidneys detect this low O2 level. That's called tissue hypoxia. Kidneys then will produce more erythropoietin, release it into the blood. EPO then travels to the bone marrow, causes the erythropoiesis to increase, and therefore the hematocrit will also increase, and that brings the level of oxygen back to normal, closes the feedback loop, and that decreases any more erythropoietin secretion. And your blood oxygen level goes back to the normal range. So because your red blood cells only live for about 4 months, they have to be constantly replaced, and the old ones have to be broken down. Erythrocytes will generally become trapped in your splenic sinusoids. Remember, your spleen is found in the upper left quadrant of your abdominal cavity. The splenic macrophages then will digest these old red blood cells, and once that occurs, the hemoglobin and then is broken down further into its constituent amino acids and iron, ions, and heme product. Heme is then converted to a waste product called biliverdin, which is responsible for the greenish pigment that you see many times when a person becomes bruised. You'll see that greenish pigment as a translucent pigment in the skin. And then it can be further converted to a yellowish waste product referred to as bilirubin. Bilirubin, of course, when it gets into the blood, it can cause a condition referred to as jaundice. And when it occurs in the eyes and the conjunctival membranes, that's called icterus. The iron ions and the amino acids are then also salvaged and recycled, and they're used to form new hemoglobin in the red marrow. Remember, nothing is wasted once that red blood cell is broken down. So first, your erythrocytes become trapped in the sinusoidic spleen, the sinusoids of the spleen. The splenic macrophages then digest the erythrocytes and then break them up. Hemoglobin then is broken down into its constituent components. First, remember, these are polypeptide chains, and so the polypeptides are broken down into their constituent amino acids, and the heme group in the middle of the polypeptide, remember, it's going to be broken down, the iron's taken out and then transferred back into the blood after it's bound to transferrin back to the bone marrow. Okay, so the iron ions and the amino acids are then recycled and used to make new hemoglobin in your red bone marrow, and then the bilirubin then is then sent to the liver for excretion. Remember, the bilirubin is then going to be added to the bile and then sent back to the duodenum via the gallbladder. Anemia is a very common condition that's defined literally as a decreased oxygen carrying capacity of the blood. There's three principal causes for anemia. One, you may just have a decreased hemoglobin. That could be from a nutritional cause or otherwise. Second, you may just have a decreased hematocrit. The red cell percentage is dropped. Okay, and the third cause for anemia is what we refer to as an abnormal hemoglobin. Either inadequate iron or something's wrong with the polypeptide units or the so you have a variety of events or conditions associated with anemia. Many of these have similar conditions and symptoms, and that reflects mainly the decreased oxygen delivery to the tissues. So anemia is kind of a constellation of conditions that occur because of one of these three etiologies, either decreased hemoglobin, a decreased hematocrit, or an abnormal hemoglobin. But the condition that applies to all of them, the main symptom is basically a decreased oxygen, and that usually causes the skin to appear what we'll call bluish in color or cyanotic. So general symptoms of anemia include pallor, which is pale skin, fatigue, tiredness, weakness, shortness of breath. Many types of anemia can also cause elevated numbers of circulating reticulocytes. The body will boost its production of EPO hormone in response to your tissue hypoxia experienced by the kidneys, that diminished oxygen carrying capacity. Severe cases of anemia can actually elevate the heart rate, cause tachycardia. And this is because the body is attempting to increase the cardiac output to match that demand for the oxygen from those oxygen-deprived tissues. But most common form of anemia is iron deficiency anemia. And that's simply caused by an inadequate dietary intake of iron. So if this is the case, sometimes it can be corrected by giving them iron supplements. Or it can be caused by reduced intestinal absorption of iron. You may have inflammation of the duodenum, of the small intestine, or even a tumor. Okay, or a slow blood loss, a chronic hemorrhaging or blood loss. Without functional hem groups, though, the erythroblasts, remember, are unable to synthesize any hemoglobin. And then you have anemia of chronic disease. That's also a common condition. And it basically develops generally as a result of another underlying etiology like cancer. It interferes with the transportation of iron, then from the liver to the red bone marrow. Other causes of anemic chronic disease are vitamin B6 deficiency, malnutrition, drug poisoning or heavy metal poisoning like lead poisoning, and even pregnancy, mainly because of increased volume that the mother has to take care of. Those all can result in a decreased hemoglobin or decreased, and we can always measure that hemoglobin levels with instruments in the lab called hemoglobinometers. So let's look at one of the causes, the decreased hematocrit. What are some of the, remember, hematocrit is the percentage of red blood cells per unit volume. So factors that can reduce the number of erythrocytes in your blood will actually lower your hematocrit. Probably the most common is blood loss, which is due to a number of causes from either acute injury, like from a car accident, or a blunt injury to the stomach, stomach ulcers, and those can lead to significant loss of red blood cells. Another cause of decreased hematocrit can be a condition called pernicious anemia. Pernicious anemia generally results from a deficiency of vitamin B12. B12 is very important for the nucleus to mature in the red blood cell, and therefore DNA synthesis to occur. So when you have a deficiency of vitamin B12, you're not going to have adequate DNA synthesis by your rapidly dividing erythroblasts, and which include your hematopoietic stem cells in your bone marrow. So many of these cases of pernicious anemia will have actually a red blood cell that's actually has a larger nucleus, and the red cell will actually appear larger, mainly because of that synthesis of those rapidly dividing cell interference. And the DNA synthesis, since it's interfered, the nucleus will generally be abnormal appearing. Erythrocyte destruction can also be caused by bacterial infections, disease of the immune system or the liver, and also lead poisoning. Those all can result in hemolytic anemia, and you can usually get evidence of this when you get a urinalysis and a complete blood count. You also have certain medications that can cause an hematocrit to fall, like ionizing radiation, and these all can inhibit or stop production of red blood cells in your red bone marrow. This condition, referred to as aplastic anemia, the cause of aplastic anemia is often not really even known. Another type of anemia is where the anemia is a result of an abnormal hemoglobin molecule. And the most common example of this is of this abnormal hemoglobin is sickle cell disease. Individuals with, let's say, a single copy of a defective gene, okay, they have what we refer to as sickle cell trait. And generally these patients are simply asymptomatic because the red cells will generally not sickle in areas of oxygen demand. But individuals with two defective copies of the gene having the sickle cell disease will produce an abnormal hemoglobin called hemoglobin S. And they will exhibit traits of sickle cell disease. So whenever the oxygen levels are low in the body, when the red blood cells are traveling through blood vessels, particularly in areas where the blood vessels branch, that's going to create turbulence, and it's also going to cause the oxygen level to be fairly low in those areas, and that causes these hemoglobin S molecules to line up and form these chains, okay, and that forms the erythrocyte, basically a sickle like you see here, okay, and it usually occurs in these smaller branching vessels. Okay, so we've discussed the red blood cells, that portion of the blood. Now let's look at their leukocytes and the immune function. Okay, remember, granulocytes are one type of leukocyte, and they're readily distinguished by their unusual nuclei. They generally have either a single nucleus composed of multiple connecting lobes. Many of these are called neutrophils, and some can be just bilobed. But all cells in this group will contain general lysosomal granules, if they're granulocytes, as well as granules unique to each type of granulocyte. And we'll discuss the three types in just a second. So the granules will divide into three categories according to what color the granules appear once they are stained. They can either stain a light lilac color, which is what you're going to see with the neutrophils, a dark purple, that's what you'll see with the basophils, a second type of granulocyte, or a bright red, which is what we'll see with the eosinophils. And they'll stain these colors when they're stained with either a basic dye or a methylene blue dye, or an acidic dye, or an eosin dye. And that, by incidentally, that's how we get the name eosinophil, which is a type of leukocyte that stains with this acidic dye. Now, your neutrophils are your most common of your leukocytes. They represent about 60 to 65% of your total white blood cells. And their granules will absorb both the acidic and the basic dyes. So that causes the granules to stain with a kind of a mixture of red and blue, which is kind of results in a light lilac color. These are very active phagocytes that ingest and destroy mainly bacterial cells, also referred to as polymorphonucleoleucocytes, or polys or PMNs. And that's because they have this uniquely shaped nucleus composed of generally 3 to 5 lobes. However, I've counted as many as 8 lobes in many of these neutrophils. So when cells become injured, they'll release chemicals, usually chemicals that are released from the damaged cell membranes of the injured tissues. And these will instantly attract more neutrophils, and that process is referred to as chemotaxis, in which the neutrophils then exit the bloodstream and release those granules into the damaged tissues. The granule contents then directly either kill the bacterial cells or attract more neutrophils and leukocytes to the region, and also help enhance the inflammatory process. Second type of granulocyte will generally have a bilobe nucleus, and these are the eosinophils. And since they take on an eosin dye and appear red, their granules appear kind of a bright red, and they're called as eosinophils. They're also phagocytes. They're slightly larger than the neutrophils, and they're generally going to ingest foreign molecules such as infections with parasitic worms and allergic reactions. So when somebody's, let's say, invaded by the agent that causes malaria, you know, the bacteria called Plasmodium vivax, which is carried by the vector mosquito, the Anopheles mosquito, the plasmodium gets into the bloodstream and will attack the red blood cells, and that's what causes the high fevers that you see in malaria. So that would be a parasitic worm attack or a parasitic bacterial attack. Also, allergic reactions are also going to have a higher percentage of eosinophils. Eosinophils in their granules also have an amount of an anti-inflammatory agent called histamine. So granules within eosinophils have many enzymes and toxins specific to parasites, but they also contain chemicals, like I said, histamine, that mediate inflammation. Now, by the way, the eosinophils represent roughly 2 to 4% of what we'll call a white blood cell differential, where you take the percentages of each individual leukocyte. Now, the basophils are the least common of your leukocytes. They represent generally less than 1% of your total white blood cells, usually between 0.5 to 1%. They generally have a bilobed or S-shaped dark purple nucleus that, because of the uptake of the methylene blue dye, you know, the basic stain. And they also contain chemicals like histamine in their granules, which mediate inflammation, similar to the eosinophils. You also see a high number of basophils in patients that have been treated with ionizing radiation, a condition that's called radiation sickness. So these are some images of neutrophils with their lilac staining granules and their nucleus with 3 lobes in this case, but they generally contain between 3 to 5 lobes. They're usually connected with this thin strand of chromatin between lobes. Remember, these represent about 60 to 65% of your total white blood cells. Notice the size of the neutrophil compared to the red blood cell. Of course, the white area is the plasma. And then here's your eosinophil with its bright red granules in its cytoplasm and its bilobed nucleus. Remember, the nucleus is dark purple because of the hematoxin stain of the nuclear material. And then the basophil which represents less than 1% of total white blood cells, generally has an S-shaped nucleus, this dark purple area that you see here, with dark purple granules in the cytoplasm which contain the histamine that help mediate inflammation. So this would be a large basophil. So those are the granulocytes. Now the other types of leukocytes are called the agranulocytes, and these include the lymphocytes and the monocytes. Both of these white blood cells will lack any granules in their cytoplasm. So that's the reason they're called agranulocytes. Now, other lymphocytes, you have B cells or B lymphocytes. B lymphocytes, when they're exposed to, let's say, an invading microbe like a virus or a bacteria, which is what we call activation, they produce antibodies, which then bind and remove the antigens from the tissues. And each population of B lymphocyte will secrete antibodies that bind only to that specific unique antigen. And we'll talk about this further when we look at immunity in the next chapter. Now, the other type of a granulocyte or lymphocyte is called the T cells, or the T lymphocytes. These are the cells that are activated also by specific antigens, which are usually, antigens on viruses or bacteria that have invaded the body. The T cells do not produce antibodies, however, but they do have membrane-bound receptors that are receptors for the individual antigens. These are made in the bone marrow, and these cells will mature in the thymus gland, particularly during childhood. So T lymphocytes will activate many of your other immune system components, and then they directly destroy many of your abnormal body cells, in particular cancer cells and virally infected cells. By the way, many cancer cells are the result of virally infected cells, like the hepatitis C virus and the papillomavirus. Remember, a virus is really a non-living entity until it invades a living, either a human body or an animal body, and then the virus, once it invades the living organism, then will use the host DNA in order to replicate and duplicate its own nucleic acid, which can be either a DNA or an RNA. So antibodies and T cell receptors will both bind to only one unique antigen based on the structure. So it's a very highly specific process that occurs. And by the way, it's these T cells, these fortified T cells that are now used for fighting certain types of cancer today, what we call immunotherapy. It's also these T cells that produce the antibodies that activate and help produce antibodies in the B cells for fighting infections by certain viruses like the coronavirus and the hepatitis virus and the AIDS virus. Now, the other agranulocyte is the monocyte. It's the largest of your leukocytes, and they generally have a large, also U-shaped nucleus, which is usually surrounded by a thin light blue or purple cytoplasm. Monocytes represent between 3 to 8% of your total white blood cells. By the way, your lymphocytes, they represent about 20 to 25% of your total white blood cells. And we'll go over that again when we look at the differential white blood count. So monocytes will only circulate in the blood briefly before they exit the capillaries to enter the tissues, where then some of these monocytes further mature into macrophages once they've been exposed to an invading antigen, which can be either a bacteria or a virus. Macrophages are extremely active phagocytic cells that generally serve to ingest dead and cells that are dying, also bacteria, antigens, and cellular debris. These are eliminated by these macrophages. They're like the garbage collectors for the human body. Macrophages also help to activate other components of your immune system, particularly in the B cells and the T cells by displaying an antigen that has been phagocytosed by the macrophage. And then it presents that antigen on its cell membrane of the macrophage to other leukocytes, like other T cells and B cells. Kind of like an identity tag, telling that lymphocyte that there's something in the body that's attacking the body. So this is basically a lymphocyte. Remember, they represent between 20 and 25% of your total white blood cells. Nucleus is generally either spherical with a thin rim of light blue cytoplasm. And of course, it becomes visible when it's stained with hematoxin stain. And then remember, lymphocytes are subdivided into B cells and T cells. B cells, remember, produce antibodies. T cells are what directly fight the invading antigen. Now, monocytes, remember, these are your largest of your white blood cells. They generally have a bilobed or U-shaped nucleus with a light blue to purple cytoplasm. Remember, these cells can sometimes mature into macrophages whenever they're exposed in the tissues to different antigens. So a very important tool for testing conditions of anemia and infection is what we'll call a complete blood count. First of all, the blood sample is drawn. It's then examined under the microscope and also by an automated analyzer. We generally have automated analyzers today. We used to have cytotechnologists that would actually count the red blood cells or the white blood cells and give a percentage. And that would take a little bit of time to do, though. Now it's fairly fast and very efficient today with these automated analyzers. And that will evaluate the number and the characteristics of the different blood cells. Red blood cells are counted in cells per milliliter, and that's then used to then calculate the hematocrit. And also, the hemoglobin concentration is also calculated, and that's a result of the hemoglobin that binds to the oxygen. And that gives you percent saturation, and that roughly tells you the concentration of the hemoglobin. So remember, both figures will give you a condition of anemia if either the red cell count or the hematocrit or the hemoglobin concentration is reduced. So some of the once the blood is drawn, then the red cell is then characterized by its size, its volume, and the concentration of the hemoglobin that's present in the cytosol. Second, the blood count and volume is also determined, as well as the number and types of leukocytes. And it's basically reported in a percentage. Now, we said erythropoiesis is basically the production of red blood cells in the bone marrow, in the red bone marrow. Now we'll look at leukopoiesis, the production of white blood cells in the red bone marrow. So leukopoiesis is a specific process in the bone marrow in which it's the same hematopoietic stem cells that produce the erythroblasts, but in this case, they're formed into leukocytes. So the hematopoietic stem cell will then divide and then basically produce two cell lines. One is the myeloid cell line, the second one is the lymphoid cell line. Your myeloid cell line produces most of your formed elements, like your basophils and neutrophils and eosinophils, and it also produces your erythrocytes and your platelets. The lymphoid cell line produces mainly the lymphoblasts, which then become committed to becoming either B cells or T cells, which then develop into prolymphocyte precursors. So first, the hematopoietic stem cell differentiates into a blast cell, which then becomes committed to becoming a monocyte, which are referred to as monoblasts. And the precursor cell that becomes a monoblast is called a promonoblast. Most all your granulocytes are derived from these monoblasts that differentiate into these precursor cells referred to as promyelocytes. You also have band cells or stat cells, which is your final precursor stage for many of your myeloid cell lines. And these then develop into mature granulocytes, like a neutrophil or a basophil or an eosinophil, that then enters the bloodstream. Now, your B cells and your T cells, which are lymphocytes, will mature in different locations. Your B cells are going to be produced and develop in your bone marrow, whereas your T cells, even though they're produced in the bone marrow, they're going to mature in the thymus gland, and that's how we get the name T for T cell. So B lymphocytes remain in the bone marrow during their entire maturation process, and your T lymphocytes will migrate from the bone marrow to the thymus gland, which is located in the lower mediastinum, to complete their maturation. But remember, the thymus gland is very active in infants and children, but it's not very active in grown adults. Once you reach adolescence, the thymus gland is starting to regress in size. So this is a figure 19.9, just basically shows you the general process of leukopoiesis, where the hematopoietic stem cell will differentiate into a myeloid or a lymphoid cell line. The myeloid cell line then either becomes a committed cell, which is either a myeloblast or a monoblast. And then the myeloblasts then develop into promyelocytes, which are basically immature leukocytes, and the monoblast develops into a promonocyte. Whenever you see the term cyte, CYTE, that usually means it's starting to mature. Whenever you see the term blast, that means it's still differentiating. It's still in the precursor stage. So the promyelocyte then will become either a neutrophil band cell, an early band cell, an eosinophil band cell, an early eosinophil, and a basophil band cell. Notice they all have this kind of ribbon or bilobe nucleus, this horseshoe-shaped nucleus. They're called band cells. So a lot of times when you get a complete blood count or a blood smear and you see a lot of these band cells for the granulocytes, you know there's a current infection going on, okay? Whereas your monoblasts, which also develop from the myeloid cell line, they then become a precursor promonocyte, which then becomes a monocyte. Now your neutrophil band cells then become mature leukocytes, which of course are going to have several lobes and neutrophils that come from the band cell neutrophil. The eosinophil will generally keep its bilobe nucleus, but have, these eosinophilic granules, these bright red staining granules, and they'll appear very bright red when you look at the blood smear. And I'll have a lot of images of eosinophils that I've taken from actual blood smears. And then your basophil band cell. These become basophils, and with their dark purple granules, which remember they can contain histamine granules. Now remember, the monoblast becomes a promonocyte. That's the precursor cell, which then matures into a monocyte. Remember, these are the ones that, when they get into the tissues, they can differentiate into macrophages. And then you have your lymphoid cell line. Remember, that's the other cell line that comes from your HSC cell, your hematopoietic stem cell. Lymphoid cell line becomes a lymphoblast. Now it's committed. Lymphoblast then becomes either a prolymphocyte, which can become either a T cell or a B cell. T cells will migrate to the bone, to the thymus gland, and become mature T lymphocytes. And the B lymphocytes will stay in the bone marrow and then become mature B lymphocytes. And then they'll migrate to the lymph nodes and the different lymphoid organs of the body, where they're going to be more freely exposed to antigens and be exposed, and you're going to have a better risk of exposure to antigens when they get to the lymph nodes, which are, remember, your lymph nodes are located more superficially in the body. Whenever you have a malignancy of any kind of blood cell, that's what we refer to as leukemias. They're cancers of blood cells or your bone marrow. They can be classified as either acute leukemia or chronic leukemia. And the reason they're classified this way is that some are aggressive or some are very slow progressing. So the acute aggressive disease progression, what we refer to as acute have a rapid increase in immature, non-functional, poorly functional blood cells. Chronic will have a slower progression, and they basically develop into a slow accumulation of abnormal mature leukocytes. Like chronic lymphocytic leukemia would be an abnormal lymphocyte, but it's generally from a chronic, slow accumulation of these abnormal leukocytes. Lymphocytic cells mainly differentiate from the lymphoid cell line. They're generally abnormal B lymphocytes. Myelogenous cell lines are from myeloid cell lines. Those can only involve any of the myeloid cells, which can be, your basophils, your eosinophils, or your neutrophils. ALL, or acute lymphocytic leukemia. It's the most common leukemia that's found in children. What happens is the abnormal cells will crowd the bone marrow. It reduces the ability for the manufacture of healthy cells. Cancer cells then start entering the bloodstream. Then they can spread or metastasize to the other tissues. The treatment for ALL, acute lymphocytic leukemia, most common in children, will vary with the type of leukemia. The generally more acute forms will metastasize earlier and require more aggressive management, whereas the platelets. Okay, remember platelets are fragments of white blood cells. Okay, they're small fragments surrounded by a plasma membrane with a few granules in the cytoplasm. They're the smallest of the formed elements. They're mainly involved in hemostasis. Hemostasis is defined as the process of controlling bleeding, stopping blood loss from an injured blood vessel. Remember, platelets are fragments of cells, so they don't have any actual nuclei or organelles that you find in your other whole body cells, like many of your other leukocytes. Platelets do have several types of granules, which contain clotting factors, enzymes. They also have some mitochondria and glycogen deposits. And that enables the platelets to carry out many of the oxidative catabolism in your coagulation process, your blood clotting. They also contain cytoskeletal elements, which include the microtubules associated with actin and myosin filaments, because we're going to see that once the platelets aggregate, they form these plugs that initially seal off a blood vessel. Once the blood vessel has basically a wall that's fractured or injured. So this is what a platelet looks like with its mitochondria and granules, glycogen granules. Here's the plasma membrane, but very small. Those are the little purple structures within the plasma. So the formation of platelets is referred to as thrombopoiesis, because the other name for a platelet is a thrombocyte. So it begins as your hematopoietic stem cell differentiates into a converted precursor cell. Those are called megakaryoblast, and these originate from the myeloid cell line. Megakaryoblast then become megakaryocytes. Remember, mega means large, karyo refers to nuclear. Okay, so they go through repeated mitotic cycles without any cytokinesis. And remember, cytokinesis is the splitting of the cell membrane and the cytoplasm. So when you have repeated cycles of mitosis without cytokinesis, you're going to have a multinucleated cell. So the cell itself never divides, and that results in this massive cell with multiple copies of DNA within a single nucleus. That's the reason they're called mega, which literally means large nucleus. Large nucleus in the cell. Cyte means cell, karyo means large or nuclear, and mega means large. So when your mature megakaryocytes are stimulated by thrombopoietin, the hormone, it then sends these extensions through the cytoplasm, these cytoplasmic extensions through the clefts in the bone marrow sinusoids into the bloodstream, and then they break off into the thousands of little platelets. And there's a video that demonstrates this process, and it's posted on Blackboard. platelets generally don't live for longer than a week. They have a limited lifespan of about 7 to 10 days, and then they have to be removed from the circulation by the liver and the spleen. So in thrombopoiesis, the making of platelets, you have a hematopoietic stem cell. Remember, this is a stem cell for all of your blood cells. Then becomes a myeloid cell line, the same cell line that differentiates into your basophils and neutrophils and eosinophils. It then becomes a megakaryoblast, which then goes through repeated mitosis without cytokinesis, so you don't get any splitting of the cytoplasm. So you get this large megakaryocyte with little arms that come out of the cytoplasm, these little cytoplasmic arms, and then they pinch out the sinusoidal capillaries and then break off when they get into the bone marrow sinusoids. And then they become platelets. So that's how thrombopoiesis occurs, or formation of platelets. Hemostasis, controlling bleeding or blood control when a vessel is injured. As we're going to see, there's 5 parts, 5 stages of hemostasis, 5 distinct events. First, they form a gelatinous blood clot that then plugs the broken vessel. But the main function of hemostasis is to limit any significant blood loss from an injured blood vessel. So the 5 stages are vascular spasm. The blood vessel basically constricts, then dilates and constricts. That's what we call vascular spasm. Second, you get a platelet plug formation. That's your temporary seal of the vessel wall. Then coagulation. This is a set of enzymes that requires 2 pathways, an intrinsic, which means within the blood, an extrinsic pathway, which occurs outside the blood. And we'll look at these pathways in a minute. And then the clot retracts, okay, and then finally thrombolysis occurs. Okay, the components of the clot have to be broken up. That's called thrombolysis. So the part 1, hemostasis, the vascular spasm, that occurs when the vessel is first injured and blood starts leaking into the extracellular fluid. Vasoconstriction, increased tissue pressure, acts to decrease the blood vessel diameter, and that will control blood loss. And your blood loss will thus be minimized as both your pressure and your blood flow are reduced locally by those 2 responses. So first, your injured vessel, your blood starts leaking out into the extracellular fluid. The vessel then goes through vasospasm, which is constriction of the vessel, which increases tissue pressure, and which decreases your blood flow and blood loss. Second event occurring is your formation of your platelet plug, which is kind of a patch, which is an aggregation of platelets, which only adheres to the injury site. And that further reduces any blood loss. So one of the things that happens is you have factors, which are really basically proteins and enzymes that are involved in this plug formation, where these collagen fibers are exposed, and then certain chemicals are released from the damaged cells of the vessel wall. Well, when the endothelial cells of the vessel wall, they'll release von Willebrand factor, VWF. And von Willebrand factor is a glycoprotein that serves to bind to different receptors on the platelet surface of the plasma membrane of the platelet. And that will then activate the platelet. But first, you have to have that exposure of the collagen from the damaged vessel wall. So the exposed collagen plus the von Willebrand factor together will make these platelets more adherent to each other, more sticky. And it'll also be more adherent to the surrounding vessel wall. So the binding of von Willebrand factor and collagen to the platelets will then trigger a series of events within the platelets, and that's referred to as platelet activation. Those platelets, once they become activated, release granules containing ATP and ADP. Remember, these are energy nucleotides, serotonin, calcium, and clotting factors. Calcium is absolutely necessary for blood clotting. The granules then attract and activate more platelets to the area. That's called basically a positive activation, positive feedback control. So first, your vessel injury exposes the collagen fibers, as you can see here, and that will produce von Willebrand factor. Von Willebrand factor then will bind to the platelets, as you can see, which then activates them and causes them to basically stick to each other, and basically also to the vessel wall. And those activated platelets then will release the contents of their granules, as you see from the image. So the contents of the platelet granules will then activate and attract more platelets to this platelet plug that's formed. Now we reach the third part of hemostasis, which is basically the coagulation cascade, or the coagulation. The process that forms the molecular glue, which binds the platelets, the endothelial cells, and all the different formed elements together. It actually is a set of two pathways, but it's a cascade of events where one event activates the other. The intrinsic pathway and the extrinsic pathway. Intrinsic pathway, because this cascade of events occurs in the blood vessels, whereas the extrinsic pathway will occur outside the blood vessel. But both these pathways will converge at the common pathway, which then leads to activating fibrin, activation of fibrin. Fibrin is going to be a thread-like material. Will act to make the platelet the plug more substantial. Fibrin is a thread-like protein that converts a soft liquid platelet plug into a more substantial solid mass. Remember, fibrin is found circulating in the plasma as fibrinogen, which is the inactive form. Remember, OGEN always means it's the inactive form of the molecule. Fibrinogen then is converted into fibrin by a series of reactions that occurs at the surface of the platelet or the damaged endothelial cells of the vessel wall. And that's referred to as the coagulation cascade. And these cascades will depend on certain enzymes, which are called clotting factors, which are synthesized in the liver. But these clotting factors are going to basically circulate in the blood at all times in their inactive form, so they don't, clot your blood vessel. Clotting factors are actually named with a Roman numeral. They're labeled with a Roman numeral in the order that they were originally discovered. So factor 2, 7, 9, and 10. 2, 7, 9, and 10. Those are all dependent on vitamin K for their synthesis. And that's the reason vitamin K is important for controlling bleeding. A lot of times surgeons will give an injection of vitamin K to their patients prior to surgery, especially when they have to do general surgery or when they expect a little bit of blood loss. So 2, 7, 9, and 10. Those are all vitamin K. They're the vitamin K dependent clotting factors. We call the intrinsic pathway because all the clotting factors that are involved are found within the blood. So the pathway is activated once the inactive protein clotting factor 12 comes in contact with the exposed collagen factors. So 12 gets exposed to collagen. Exposed collagen then activates 12 to active 12. Whenever you see the A in front of the Roman numeral 12, that means it's the active form. Factors 11 and 9 then become activated from 12A. So 12A is an enzyme that activates 11. 11A then subsequently activates 9. So it goes 12A to 11. 11A then activates 9, and then it basically forms this coagulation cascade where each factor, once activated, then activates the next factor in the pathway. Factors 9A and 8A plus calcium, these form an enzyme complex that activates factor 10 to active 10, 10A, and that completes the pathway. And that's called the intrinsic pathway because it occurs within the blood. So first, in your intrinsic pathway, exposed collagen fibers will activate factor 12 to 12A. There's your exposed collagen. There's your platelet plug. There's your factor 12, which is inactive. Then active once it's exposed to these collagen fibers. Then factors 9 and 11 become activated. 11 and 9 become activated through activation of 12A. Factors 9A and 8A and calcium form an enzyme complex. This is an enzyme complex, this area that I'm circling here. And that will then activate factor 10 to active 10. Okay, and that's called the intrinsic pathway. Now, the extrinsic pathway occurs outside the blood, but occurs simultaneously with the intrinsic pathway. So what happens in the extrinsic pathway outside the blood vessel, the damaged subendothelial cells will display a protein referred to as tissue factor. Tissue factor then activates factor 7 to 7A, which is the active form. So factor 7A, tissue factor, and calcium forms another enzyme complex, which then activates factor 10 to active 10, 10A. So first, your subendothelial cells display tissue factor, as you can see here. These are your subendothelial cells. Here's your vessel. Okay. Then the tissue factor then activates factor 7. 7 is activated. And then factor 7A, tissue factor, and calcium form this enzyme complex that then further activates factor 10 to 10A. And that's what we refer to as the extrinsic pathway because it occurs with exposure to these subendothelial cells displaying tissue factor, which is out in your tissue, not inside the blood. So it's called the extrinsic pathway. So factors 10A and 5A and calcium then form prothrombin activator, which then converts prothrombin into thrombin. So here's your factors 10A, 5A, and calcium. Okay, and they then form prothrombin activator, which then converts prothrombin into thrombin. And thrombin in turn then converts fibrinogen into fibrin. And fibrin becomes associated with the platelets, and that forms the thread-like protein that kind of molecularly glues everything together. And that's what you see here in a scanning electron micrograph, where you see the red blood cells becoming trapped within inside the fibrin threads and the different platelets and leukocytes. And that's what you see when the blood is first coagulated. So after the coagulation cascade, then you'll have the last portions of the coagulation. Okay, what's the best way to remember it? Remember, 3 goals are going to occur in coagulation. First, you have to produce factor 10A. Remember, that's sometimes called the common pathway because it's the goal of both the extrinsic and the intrinsic pathways to produce active enzyme factor 10A. Even though the reactions are different in each pathway, the end result is the same. Okay. Second goal is to produce thrombin, which is also another product of both the intrinsic and extrinsic pathways. Factor 10A will accomplish the second goal of coagulation by reducing the enzyme thrombin, and that occurs in the common pathway because it's common to both the intrinsic and extrinsic pathways. And then the third goal is to produce fibrin. Okay, remember, thrombin in turn will accomplish the third goal of coagulation, which is produce fibrin from fibrinogen, basically to hold the platelet plug together and then seal the wound. And that's the final result of your common pathway. Claw retraction, that's the fourth part of hemostasis, the fourth event. That occurs as the coagulation cascade nears its completion. That involves your motor proteins, actin and myosin fibers. That helps the involved platelets contract. That helps bring the edges of the wounded vessel closer together. And serum is referred to as a fluid consisting of plasma without the clotting factors, the clotting proteins. And that's the serum is then forced out of the clot during retraction of the clot. That's that clear fluid that's exuded out of an injured blood vessel. So that's what we refer to as clot retraction, and that helps bring the edges of the wound closer together. And the serum is then squeezed out of the clot. And that brings us to our last stage, which is thrombolysis, which is a process that begins after the injury is healed and blood clotting is no longer necessary. So fibrinolysis, which literally means breakdown of fibrin. And that's the process that breaks down the fibrin glue. Endothelial cells release a tissue plasminogen activator, TPA, the same TPA that's used in it's given into coronary artery blood clots that occur also that involve TPA that's given by paramedics during a myocardial infarction when you have a coronary artery that's blocked by a thrombus. So it initiates thrombolysis, literally the breaking down of the thrombus. Urokinase is very similar to TPA, tissue plasminogen activator, but it's produced by the kidneys. It's found in both the plasma as well as the extracellular fluid, and they can also initiate thrombolysis, the breakdown of thrombin. TPA activates plasminogen, which is the inactive plasminogen is found in your plasma. It then binds to fibrin molecules as they become incorporated into the blood clot. TPA then converts the bound inactive plasminogen to the active enzyme plasmin, and plasmin in turn degrades fibrin and the clot dissolves. The remaining components of the clot then dissociate from the endothelium, and you finish your process of coagulation, and your hemostasis is ended. So first, the endothelial cells release the tissue plasminogen activator, TPA. Okay. TPA then activates the plasminogen to become plasmin. Plasmin then degrades fibrin, and the clot then dissolves, as you can see here. So this is kind of a summary of all of the events that we've talked about. Okay, vascular spasm, where the blood flow through the injured vessel decreases through the constriction of the vessel. Platelet plug formation, that seals the vessel wall. So just a temporary plug, though. Then you get your coagulation, which consists of the simultaneous intrinsic and extrinsic pathways, which produce factor 10A, as you can see. This would be the intrinsic pathway, which involves 9 and 7 in calcium, and this is the extrinsic. Remember, the intrinsic occurs in the blood, and the extrinsic occurs in the tissue. It involves the subendothelial factor, but they both converge and convert 10 to 10A. And then you get your the common pathway producing thrombin, where prothrombin is converted to thrombin from the combination of 10A, 5A, and the enzyme complex and plus calcium. Okay, then the coagulation, where the thrombin converts fibrinogen into fibrin, and the platelet plug is then glued together. It's more of a substantial clot now. Then you get retraction of the clot, which brings the edges of the wound closer together. It also helps express the serum. And then you get thrombolysis, where the plasmin degrades the fibrin, and the clot then dissolves, as you can see here. You see no more clot here. This is a table 19.2 that gives you each clotting factor labeled with their appropriate Roman numeral, their alternate name. So factor 1 is actually fibrinogen, made in the liver and platelets. Remember, fibrinogen becomes fibrin, the sticky protein that threads, that glue the platelet plug together in the common pathway. And there's prothrombin, which is actually factor 2. Factor 3, which is tissue factor. Factor 4 is calcium ion. Remember, calcium is important in activating factor 10 and prothrombin. Factor 5 is also called labile factor. Also another plasma protein made in the liver, but stored in platelets. Factor 7, also called stable factor. Also made in the liver. Synthesis requires vitamin K. Remember 2, 7, 9, and 10 require vitamin K. Factor 8 is called the antihemophilic factor A. Factor 9 is also called Christmas factor. Remember, all these are plasma proteins made by the liver. Remember, 9 is also dependent on vitamin K. 10 is also called Stewart factor, also dependent on vitamin K. 11 is also called PTA, plasma thromboplastin antecedent. It's a plasma protein made by the liver and stored in the platelets. And then factor 12 is also called Hageman's factor. And factor 13 is also called fibrin stabilizing factor. Activated by fibrin and stabilizes the fibrin mesh. Okay, so clotting is regulated. Blood clotting is another example of positive feedback mechanism. It's tightly regulated mainly to prevent any kind of mishaps. You wouldn't want any blood clots occurring in any of your vessels that could potentially cause either a stroke or a blockage of a coronary artery, which could end result could be, of course, a myocardial infarction or a stroke. So you don't want any kind of it has to be tightly regulated so that you don't get any unformed or clots that have been formed to not completely be dissolved. So the endothelial cells in your vessel wall will produce and secrete two chemicals. And these chemicals can regulate your first and second stages of clot formation. The vasospasm and the platelet plug and the coagulation. So prostacyclin is a prostaglandin that inhibits aggregation of platelets. And nitric oxide, also made by the endothelial cells that line your vessel wall, causes vasodilation. And that serves to do the opposite of vasoconstriction. Also, your endothelial cells and your liver cells, your hepatocytes, produce anticoagulants, which inhibit coagulation. A very important anticoagulant is antithrombin 3, AT3. It's a protein that binds and inhibits the activity of both factor 10A and thrombin. It also prevents activation of new thrombin. When they're trying to prolong bleeding or prevent blood clots, a lot of times they'll give them antithrombin 3. Heparin sulfate. This is a polysaccharide that enhances antithrombin activity, and that's how it extends bleeding times. A normal bleeding time is, between 2 to 3 minutes, as long as maybe 4 minutes. So heparin can extend bleeding times up to 10 minutes, sometimes longer. And that will prevent many types of blood clots from forming that could be occur from, let's say, an irregular atrial beat, like you see in atrial fibrillation. And the third anticoagulant, which inhibits coagulation, is protein C. When activated by protein S, it can catalyze reactions that degrade factors 5A and 8A. Remember, these are the clotting factors. So these are all thrombolytic agents, and all of these are made by different parts of the body, which help regulate the formation of clots. And these are all summarized in this table 19.3. Now let's look at disorders of clotting. This is a condition in which the clotting is not properly regulated. Of course, that can create drastic consequences for maintaining homeostasis. There's two types of clotting disorders. First, you have bleeding disorders like hemorrhaging, and second is called hypercoagulable conditions, like when somebody is septic. Okay. Bleeding disorders, these can increase blood loss from even minor injuries because the blood is not able to clot. And it's secondary usually to a clotting protein deficiency Hemophilia A, also called classical hemophilia, is caused by a shortage of factor VIII. This is the type of hemophilia that occurred in Queen Victoria in her family, which included the German Chancellor Wilmark during the Chancellor of Germany before World War One. Hemophilia B is caused by a shortage of factor IX. And basically these are hereditary usually. And then the second type of clotting disorder is called a hypercoagulable condition, in which clotting is more readily. And that results in the formation of an inappropriate clot, and that's referred to as thrombosis. A clot or a thrombus is extremely dangerous. It can obstruct blood flow through a blood vessel. Thromboembolism can break off of a thrombus and then include a smaller vessel downstream. Thrombi commonly form in your deep veins of your legs. That condition is referred to as deep vein thrombosis. Pulmonary embolism is probably the most dangerous complication of a deep venous thrombosis, where the emboli break off the thrombi in the legs and lodge in small blood vessels in the lungs. You treat thrombi or emboli with drugs that help prevent the clotting process, and you do that mainly with anticoagulants. Heparin and the derivatives of heparin, which are mainly used in-house in hospitals, the action is nearly immediate, but it has to be injected, so these can be done on an outpatient basis. The other anticoagulant is also widely used, but usually as an outpatient, is called warfarin or Coumadin. It used to be formerly the main ingredient in rat poison. It's used for outpatients. It can be given orally, and it also inhibits production of all the vitamin K-dependent clotting factors by the liver, 2, 7, 9, and 12. It does not affect factors, though, already present in the plasma, and it generally takes 2 to 3 days for any noticeable effect. So when somebody is being staged for having an anticoagulant therapy, they'll probably put him on heparin while he's in-house in the hospital for about 1, 2, or 3 days, and then they'll discharge him home on Coumadin. Other antiplatelet drugs, which also help prevent clots, are aspirin. Aspirin actually inhibits the enzymes that contribute to platelet aggregation. Remember, aspirin is an acid, orthoacetylsalicylic acid. Clopidogrel, also referred to as Plavix. It blocks the receptors on the platelets and therefore inhibits platelet activation, also called the glycoprotein 2B and 3A inhibitors. These are also used in heart attack patients who also work similarly. These glycoprotein 2A and 3B inhibitors used in heart attack MI patients work very similarly. And then you have your thrombolytic agents, TPA or urokinase. What these do is restore blood flow rapidly to prevent any kind of tissue damage whenever the thrombi or emboli have either caused a stroke or a heart attack. So when a person's having an MI, they get him into the ER or a paramedic within 20 minutes. They can give him TPA, along usually with heparin, and they can generally see the clot dissolve, and the EKG will also resolve to normal. Usually, if you see ST changes in the EKG, which is evidence of usually an MI, that sometimes will stabilize, and the ST segment won't be elevated. It'll go back to baseline after the TPA is administered. Blood typing and blood matching. Blood typing and matching. Okay, blood transfusions. These are when blood is taken from a blood donor and then given to a blood recipient. It's commonly used treatment modality used in today's medicine, but that's not always been the case. So when we discovered surface markers or antigens on red blood cells, also found on all biological molecules in all cells, including your red blood cells. And that then led to more safer transfusion practices, even though we've done transfusions for many, many years. Remember, your immune system will recognize foreign antigens. Remember, antigen is generally a small molecule present on any virus or bacterial that the body recognizes as foreign. And it responds by trying to remove those antigens. And this response was responsible for many of the early fatalities in the early blood transfusion patients. Remember, antigens on red blood cells are genetically determined carbohydrate groups on the red cell membrane, which give rise to the nearly 8 different blood groups. 2 groups of the 30 different antigens are found on your red blood cells that are particularly useful for clinical use. These are called the ABO blood group and the RH blood group. So if you have 2 antigens, one A and one B antigen, and you basically have 4 ABO blood types. So if you have type A blood, that means you only have the A antigen present on your red blood cell. If you have type B, that means you only have the B antigen present on your red blood cell. If you have type AB, that means you have both the A and the B antigens on your red blood cell. And if you're type O, that means you have neither A or B antigen present on your red blood cell. Remember, there is no O antigen. O just simply denotes the absence of either A or B antigen. So if you were type O, that means you would have both recessive alleles for both the A antigen and the B antigen. Now, the RH blood group features the RH antigen, which is first discovered in the rhesus monkey, and that's how we got the name RH on their red blood cells. So if you have the RH antigen, that means you're RH positive, and those without the antigen are called RH negative, also called the D antigen. The ABO and the RH blood groups will be combined, will give rise to 8 common blood types. Type O positive is the most common blood type in the United States. I think it represents about 48%. AB negative is the least common type of blood type. And that's less than 1%. So blood typing is done in the laboratory utilizing antibodies that can bind to individual antigens on red blood cells. Antibodies, these are also called agglutinins because they bind to surface-bound antigens, causing them to clump together, or agglutinate. So that's why they're also called agglutinins. Ultimately, agglutination promotes red blood cell destruction, and that's called hemolysis. So here's an erythrocyte that has A antigens, as you can see by these A antigens, these little purple or the white letter A in the circle. Okay, those are the A antigens. So this person has A blood. Okay, and then the antibodies. These are called A antibodies. Antibodies are Y-shaped molecules, like you see here. Okay, so the anti-A antibodies will bind to the A antigens, as you can see here. Okay, the Y-shaped form on the antibody binds to the A antigen on the red blood cell, and that causes these red blood cells to clump or agglutinate. They aggregate together, and these agglutinated erythrocytes then eventually undergo hemolysis. So that's why when you mistype blood types, they can be fatal. So how do we do blood typing? Well, one is the blood sample is treated with 3 different antibodies. Remember, agglutination or clumping indicates that the antigen is present on the red blood cells. No agglutination indicates that the specific antigen is absent from the red blood cell. We use 3 different antibodies. Anti-A antibodies will bind and agglutinate A antigens. Anti-B antibodies bind and agglutinate B antigens, as you see in this image here. And anti-RH or anti-D antibodies bind and agglutinate RH antibodies, as you can see in this image. So here you've got type A blood. This is agglutination, so this person must have the A antigen to agglutinate with these anti-A antibodies that are added to his serum, his blood. Whereas this person, who has type A blood, has been given anti-B serum where the antibody is a glutenate B antigen. Since he doesn't have the B antigen, you don't see agglutination in this well. So this person had to have type A blood because it agglutinated his A antigens. On the other hand, this person, type B blood, would have the anti-B antibodies agglutinating the B antigen on his red cell membrane, whereas the A antibodies would not agglutinate because he doesn't have the A antibody, the A antigen. AB blood type, you'd see agglutination in both wells because he has both the A and the B antigens agglutinating with both the A and the B antibodies, whereas type O has no antigens on his red cell membrane. So you don't see agglutination in either well. You gave anti-A antibody in this well, you gave anti-B antibody in this well, but no agglutination. That means he doesn't have either antigen, so he's type O. If you also gave the Rh antibody, you could also, we just call, if you do have the Rh antigen, we just call you Rh positive. So if you had agglutination over here with the Rh antibody, if you had agglutination, which you would see clumping just like you see here, he'd have A positive blood. But they don't test for the Rh antibody and the Rh antigen. So remember, blood samples from 4 patients are combined with antibodies. Agglutination indicates that a specific antigen is present on that person's red blood cells. Remember, in blood transfusions, your immune system recognizes antigens on your red blood cells as self-antigens and does not produce antibodies to self-antigens. Because if it did, your antibodies would bind your own antigens. Your immune system does produce antibodies to foreign antigens. That means antibodies are present in your plasma only if antigens are absent from your red blood cells. And this is basically produced in your in the early embryo. Remember that anti-A and anti-B antibodies are preformed. They are present in the plasma, even if the individual has never been exposed to those antigens. Whereas anti-Rh antibodies are produced only if a person has been exposed to blood containing Rh antigens. Therefore, an Rh negative individual, which means he has no Rh antigen on his red cell membrane, generally has no anti-Rh antibodies unless he or she has been exposed, or what we call sensitized, to positive Rh red blood cells. And we'll see an example of that in a condition that we call erythroblastosis fetalis, or hemolytic disease of the newborn. So we use antigens and antibodies as our basis for blood matching. Where blood is taken from a donor, then it's screened for compatibility prior to its administration to a recipient. A match will occur if the donor blood type is compatible with the recipient blood type. Transfusion reactions is when the recipient antibodies will bind to the donor antigens, which then ultimately causes agglutination that destroys the donor red blood cells, and it can possibly then lead to kidney failure and then finally death. When these agglutinated blood cells start to clog up the kidney tubules, that will lead to kidney failure. So hemolytic disease of the newborn, also called erythroblastosis fetalis, it only occurs when an Rh negative mother gives birth to a positive Rh fetus. So during birth, the fetal red blood cells will enter the mother's blood, usually during the vaginal delivery of the baby. And that stimulates then her immune system to produce anti-Rh antibodies. First pregnancy generally is typically not at risk, okay? But in subsequent pregnancies, the maternal anti-Rh antibodies then can cross the placenta and then immobilize any positive Rh fetal red blood cells. So if a mother's Rh negative and she has a second child that's also Rh negative, you wouldn't have this disease. But if she has another baby that's positive for the Rh factor, then you would probably have erythroblastosis fetalis. We now can effectively prevent hemolytic disease of the newborn by blood type screening. And if the woman is Rh negative, she can also be given Rh immunoglobulin, okay, also called RhoGAM, okay, RHOGAM, which does contain anti-Rh antibodies that bind the fetal red blood cells in the maternal circulation that might have entered. And that prevents any maternal production of anti-Rh antibodies. They generally give them 3, the mother that's Rh negative, and she is known to have be married to, let's say, a positive Rh partner. Then they could have a positive Rh baby, and they usually give them RhoGAM 3 times in her pregnancy. So this is an example of, in this first case, you've got a blood match where you got donor type has O negative blood. Remember, he doesn't have any antigens on his red cell membrane. Okay. Then he's given to a recipient that has type AB positive blood. Okay, so he's not going to have any antibodies in his serum. Okay, therefore you get no agglutination with the red blood cells. Okay, so that's a blood match. When you basically take an O negative donor and give to an AB recipient, AB positive recipient. By the way, the O negative is basically what we refer to as the universal donor. And type AB positive is what we refer to as the universal recipient, because they receive any person's blood type. Whereas type O negative blood cannot receive any blood type, but they can donate to anybody. Now, this second illustration illustrates a blood mismatch. Here, in this case, the donor is type AB positive. He has A, B, and the Rh antigen. And then it's given to a recipient that's O negative. Okay, he's type O negative blood. So he's going to have antibodies to anti-A, anti-B, and Rh antibody. Okay, so therefore, you're going to have agglutination because he has all 3 of these antigens that each of these antibodies can react with, as you can see here. Okay, the A is reacting with the A antibody, the Rh antibody is reacting with the Rh antigen, and then the B antibody is reacting with the B antigen. And that will cause a blood mismatch, and that's going to cause a transfusion reaction. So blood type O negative is referred to as a universal donor blood type because their red blood cells do not have either the A, B, or the Rh surface antigens. They can be given to any blood type in an emergency when blood matching may be not an option. The universal recipient is blood type AB positive, and that's because if you're AB positive, you won't have any antibodies in your serum. So those individuals do not make any antibodies to either A, B, or Rh antigens, so they can receive blood from anybody. Remember, matching is still the safest practice. Remember, the transfusion reaction is a reaction between the donor red blood cell antigens with the recipient's antibodies. Okay, that's what causes the transfusion reactions. It's the donor's red blood cells antigens, not the donor's antibodies that you worry about. It's only the donor red blood cell antigens that you worry about. This is the 8 major blood types, showing you the prevalence in the United States population, the antigens present on the red cell membrane, the antibodies present in the plasma. This is who they can receive blood from. Remember, O negative is the universal donor. So they can donate to all blood types, but they only can receive blood from their own blood type. Whereas AB positive, this one up here, they can receive blood from all blood types, okay? But they can donate to their own blood type, only their own blood type. And that's the reason they're called the universal recipient. So what about the donor's antibodies? Remember, donor antibodies can bind to recipient's antigens, and unless their blood types are exactly matched, some antibodies in the donor might destroy a few of the red blood cells in the recipient. So if you look at this example here, Ed's 100 antibodies might destroy 100 of Tom's red blood cells. But remember, Tom has received millions of new red blood cells from Ed, so he won't really miss 100 red blood cells. This is the second example. You see donated AB red blood cells from Tom. Remember, there's no major erythrocyte antibodies from Tom because he's AB positive. And then you have Ed's O negative red blood cells. Remember, there's thousands to millions of Ed's anti-A, anti-B, and anti-RH antibodies. So as Ed's leukocytes come into contact with Tom's red blood cells, Ed's immune system makes millions more of those antibodies. That results in a transfusion reaction, which Ed's antibodies destroy every one of Tom's donated red blood cells. Remember, worry about the donor's antigens and the recipient's antibodies. That's how you can predict whether there's going to be a blood match or a blood transfusion reaction, a mismatch.